All right, everyone. Thank you so much for coming. It's really my pleasure to introduce you to Dave Schaffer, a professor at the University of California, Berkeley. Professor Schaffer is a professor of chemical and biomolecular engineering, as well as bioengineering and neuroscience. And the current director of the Berkeley Stem Cell Center, previous director of Berkeley Stem Cell Center. That's great. I can give you a walk-through introduction. I'll give you some highlights that are kind of close to my heart. Maybe I can just sort of say a little bit about Dave's trajectory, starting at Stanford University, being an undergraduate in chemical engineering, moving on to MIT to do a PhD in chemical engineering. Finally ending up sort of unusually in Fred Gage's lab at Salk, and Fred Gage is actually a very well-known neurobiologist, also a bit unconventional with interesting unconventional approaches, which I like very much. And then after that, they started at Berkeley as assistant professor, where he's been ever since, kind of at the intersection of chemical and chemical engineering, bioengineering, and stem cell biology, as well as sort of gene therapy and genetic approaches as well in cells. What I know Dave for is his work in stem cells. Obviously, he's one of the pioneers in using stem cells to build three-dimensional structures, specifically using materials that were also developed in Dave's lab. I will say personally that I use Dave's products. One of my most important papers was published based on Dave's material that they built in the lab. And he's also been one of the pioneers in using optogenetics to drive stem cell signaling back in 2013, right? That's quite a huge accomplishment. He also has a lot of interest in using stem cells to understand early development, neural development as well. However, we won't hear any of that today, but instead you'll hear Dave's tremendous efforts in advancing gene therapy approaches. So please.
Thank you so much for the very kind introduction, and terrific to be here visiting all of you. It's my first time on this part of the Columbia campus. I've been up to the medical school beforehand, but it's terrific to see what a pretty gorgeous, vibrant environment this is. Wonderful invitation. And I also want to encourage people, if anybody has any questions while I'm speaking, please don't hesitate to raise your hand. It's kind of fun to have a two-way discussion. So we work in two major areas, as was described, genomes as medicines or cell therapy, and genes as medicines or gene therapy. And today I'm going to be talking about the second of those two efforts and our efforts to be able to get into the clinic. But for both of these cases, what really drives me is this list of issues or problems that are unaddressed in human health. So regardless of which tissue you look at, and let's pick on the nervous system for a moment, we have both complex diseases, including Parkinson's and Alzheimer's, where you can't really pin the blame on specific genes. And there are also so-called monogenic diseases, where you can sequence the genome, understand which lesion or which mutation results in the development of disease. But regardless of which of these two situations it is, these conditions result in long-term degeneration of a tissue, loss of function, loss of quality of life for patients, in some cases, loss of their life. So there's a very strong interest and need to be able to develop novel therapies to treat these conditions. And to do so, we can turn to a very basic central tenet of biology, which describes the information flow and the flow of function throughout human systems or biological systems. Information is, of course, stored at the level of DNA, at the level of AGTC. This information is transcribed into RNA and then translated into a protein, of course. And from the point of view of somebody who's interested in treating a disease, any of these levels of information can be viewed as a drug target, right? So the majority of pharmaceuticals that are currently being developed or utilized within the clinic are actually targeting at the level of protein. So small molecules float around, they find an active site of an enzyme, typically block it. Monoclonal antibodies float around on the outside of the cell, find a target and block its function. So it is possible to move to a different level and use RNA as a drug target. So, for example, companies like Alnylam and Ionis are using RNAi and antisense RNA to be able to target RNA as a drug. And furthermore, we all know, hopefully so, that messenger RNA itself can actually be a therapeutic. Thank you, Moderna and Pfizer, right? So we're interested in targeting at another level though, which is at the level of DNA. So if you're delivering a gene, it's called a gene therapy. If you're delivering a cell, it's a genome therapy. If you're changing the sequence of DNA, it's a genome edit. So the reason that these are very powerful, targeting at the level of DNA, is that proteins and mRNAs and small molecules are temporary residents of the body, right? They turn over, they have a finite half-life. DNA can become a permanent part of an organ or a tissue, so you can begin to think about both treating the underlying cause of a disease, as well as single administration, long-term therapeutic benefit, one and done, ideally or potentially. And so you can illustrate that idea by considering timescales. So by the time, between the time that somebody is diagnosed with Parkinson's typically and when they pass away is around 15 to 20 years or so. Congestive heart failure and age-related macular degeneration, typically around a decade. The half-life of small molecule, hours. If you move up to a protein like a monoclonal antibody, it improves to days, but by definition, these are not cures, right? They're treatments. Whereas if you deliver DNA as a therapeutic, and I'll be describing these clinical trials, single administration back in the 2000s have resulted in long-term therapeutic benefit that these patients are still enjoying. Okay, so there's a strong interest in being able to create DNA as a medicine. However, it's very challenging to be able to do so. So in particular, I'm going to talk about our efforts to engineer biologics, or the field as a whole, where a biologic is something like a protein or a virus or a recombinant DNA molecule. So if you're trying to engineer an antibody, and this is what made Genentech famous as a company, they made antibodies, you're really focusing in on engineering just the tips of this molecule, these small regions that bind to the target. So that's a limited, constrained problem. Or maybe you actually want to engineer a brand new protein that can bind to a target. And this is work that came out of David Baker's lab, in which he used a computational tool known as Rosetta to design a protein binder from scratch. And this particular protein bound to the receptor for SARS, and it actually served as a picomolar inhibitor of SARS's capacity, or SARS-CoV-2, to infect a cell. So in other words, the field has advanced to the point where you can computationally design at least a small protein, this is around 8,000 molecular weight, from scratch and get really interesting properties out of it. By comparison, DNA is really difficult to actually engineer. So this is the virus that really has been the focus for half of my career. And unlike this one, which is relatively small, this virus is 4 million in molecular weight. It has protein inside of it. It has DNA inside of it. And you have to manufacture it, which is really tough because you have to get large numbers of plasmids inside of cells, which is difficult to do at scale. And then you want to deliver it so it drops off its DNA medicine inside the cells of a patient, trying to deliver DNA medicine to treat a disease. And so it has to be administered, biodistributed through the right part of the body. For example, find heart tissue, if that's where you would like to deliver DNA. Has to pass through that tissue. As chemical engineers, we think about flow through packed beds. So it's difficult to get this large particle deeply inside of a tissue. Then it reaches the surface of a cell, and it has to be able to traffic its way through the cell into the nucleus until it drops off the DNA at its site of interest, at the site of activity. And then finally, that DNA itself, which is the final drug product, is complicated, much more complicated than an antibody, because it contains regulatory elements that control how much messenger RNA is produced. So this is a really complicated problem compared to engineering one of these. And so we became interested in engineering these viruses to be able to make them better at carrying DNA inside of cells, but we're daunted by the complexity of the problem. That's a significant challenge within the field. So I'd like to talk in particular about the virus that we've been utilizing for half of my career. It's not SARS-CoV-2 or Ebola or West Nile or HIV. It's actually not even a pathogen. It doesn't cause human disease. So it's this harmless ball of proteins called the Adeno-Associated virus, or AAV. The virus only contains 2 genes, the first of which mediates or causes the replication of the viral DNA. And the second encodes some proteins, 60 copies of which self-assemble to form the soccer ball. And this Haller soccer ball, after it assembles, actually gets loaded with a copy of this DNA because it actually has a pore right here at the 5-fold axis of symmetry. And as a result, it creates an infectious virus. So it's among the smallest of all viruses. It's non-pathogenic. A number of different variants have been isolated, and they have differences in their amino acid composition on the surface, which raises the idea that by engineering the surface of this virus, maybe it could change its delivery properties. So you can convert it into a gene delivery vehicle by simply cutting out the viral DNA, pasting it onto a separate piece of DNA, and that liberates space for you to insert your DNA medicine, your gene of interest. And this gene of interest will then in turn get loaded inside of this particle, and this particle then serves effectively as a Trojan horse to carry DNA medicine inside of cells rather than carrying the viral DNA. So this has a number of advantageous properties. 90% of you have already been exposed to one or more natural versions of AAV, probably since the time you were going to daycare. And it's non-pathogenic, so it didn't harm you, you didn't even notice it. It's somewhat efficient. It is being used to carry DNA medicines inside of cells in clinical trials involving muscle, liver, lung, brain, and retinal disease. After depositing its DNA in the nucleus of a target cell, the DNA sticks around for a long period of time, which is what we want for one and done. And in addition, there still needs to be progress in manufacturing. This is a significant challenge, but the field is getting better at that. And the net result is that there are actually 2 FDA-approved gene therapies based upon these viruses. So this one is by Novartis. It's a therapy called Zolgensma for SMA, spinal muscular atrophy, which is the leading inherited cause of infant death. Patients have a median lifespan of 10 months. And patients who are treated with this back in 2014 are still alive. In addition, this is a drug or a product developed by Roche Pharmaceuticals called Luxturna, and I'll show you a movie of it in a few moments, but it's designed to be able to treat a blinding condition. And then finally, hemophilia B, which I'm sure you're familiar with, is a bleeding disorder caused by defects in the blood clotting factor. A company called Unicure has submitted an FDA approval application to get a gene therapy approved for this. It's anticipated that'll happen within the next 6 months or so. So for this drug, Luxturna, here's some of the key clinical data that these patients are unlucky enough to have inherited from both of their parents a defective copy of a gene, which is important for the function of photoreceptors. So in the absence of this gene, with these mutations, patients progressively go blind. And as you can see here, this patient has a hard time even making it through a relatively simple maze. So investigators took the correct copy of the gene, the DNA, loaded it into the virus, injected it into the patient's retina, and you can see 6 months later, the results very simply speak for themselves. So this became the very first clinically approved or FDA-approved gene therapy for a rare disorder, which happened in 2017. Okay, so that is the good news. You know, the field is starting to work, but there's some challenges and problems. And these are all delivery problems. They're, if you will, mass transfer issues. So the first of these is that I referred to, a lot of us have been exposed to this virus. We've been accidentally vaccinated against it. We have high concentrations of antibodies waiting to neutralize and take out these viruses. In addition, the virus biodistributes to the wrong tissues inside the body. Lots of it goes to the liver, not very much of it goes to other tissues where you may want it to go. It has inefficient spreads within that target tissue. Again, this is the packed bed problem. It has an complete inability to target delivery to some cells, but not others. And then finally, with most cell types, it's very inefficiently taken up into those cells. So all of these, results in significant challenges when we try to extend the field beyond those 2 initial successes. So you might ask the question, how did you get FDA approvals in the first place? So these investigators really identified low-hanging fruit, diseases that were easier for one reason or another to treat, and they brute forced the delivery. So in these cases, with Zolgensma and the hemophilia B product, we used massive doses within the patient to overcome the fact these viruses aren't very efficient. And in the case of Luxturna, as I'll describe in a second, it requires very invasive surgery to put the virus right next to the cell target. Otherwise, it would have no hope of getting it. Okay, so the field has been trying to deliver DNA to other tissues other than the liver or the retina. And they have to contend with the fact that most of these viruses, really like going to the liver. They're not very efficient at infecting it, but they like distributing there. And so to be able to overcome this distribution problem, there was a company called Astellas that was trying to treat a condition that was fatal in young boys. So it was a worthy endeavor. And they chose a version of AAV called AAV8. There are about 100 different versions of natural versions. And for every single particle of AAV that goes to the heart, 1,000 go to the liver. So they compensated for that and tried to get enough delivery to the heart by simply jacking up the dose extremely high, to the point where they were delivering 10 to the 16th of these particles, 100 copies of this virus per every single cell in the body. And this virus is pretty stealthy. Like I mentioned, you've all been exposed to it, never noticed it. It doesn't really cause much inflammation, has very mild interactions with the immune system. But if you stick in, 100 copies per every single cell in the body, it can awaken the immune system. So unfortunately, in this trial, 4 boys died of this gene therapy because their immune system detected the virus, panicked, set off inflammatory responses that resulted in the loss of the patient. So in other words, viruses never evolved in nature for our convenience to use as medicines. And if we would like to utilize them for this disease or many others, it's quite simply incumbent on us to improve the properties of the virus, to make them much more efficient, more targeted, etc. Okay, so we'd like to re-engineer these viruses, but we have this problem. Viruses are incredibly complicated. I described how complex these systems were. So at the time that we began to work in the field, investigators were studying mechanistically the pathway that these viruses would take to be able to go from outside of the cell to the inside. And then we're trying to rationally engineer new versions of these viruses, kind of one amino acid at a time, to be able to do that. But this is 4 million molecular weight. This is a really complicated entity to be able to engineer. More often than not, when they made changes to it, they crashed the system. They completely eliminated the ability of the virus to deliver DNA. So this is back in the 90s when I started on my lab in Berkeley, and there was a brilliant chemical engineer that we all know, Frances Arnold, who had started up her program at Caltech, and back in the early 1990s began to develop a new solution for engineering complicated biomolecules. So what was done in the enzyme engineering field, which is where Frances works, is that they would stare at a crystal structure, hypothesize an amino acid change it, express the protein, characterize it. Maybe they got a desirable change to the protein. More often than not, they didn't. They would have to go back to the drawing board, create another variant. So what Frances did was to emulate the way that nature created proteins in the first place, which is to create a large number of them, genetically diversified pool, and then perform a selection to be able to identify and pick things. And this could be a very effective approach in situations where you don't know enough about how to do the rational design, but it can lead to significant future insights about the mechanism of how systems work. So Frances coined this process of iterative genetic diversification and selection as directed evolution, and she, of course, won the Nobel Prize in Chemistry for this work in 2018. So we were the first to bring directed evolution into the gene therapy field. And the way that it works is that we start out with a number of natural versions of the virus. I mentioned there are 100 different natural AAVs, and each of which may be an interesting starting point to be able to do some evolution. And then we have genetically diversified these using 42 different approaches to date. A lot of rational thought went into this, a lot of computation, a lot of modeling to enable us to design optimal libraries. I'm not going to go into that today for the purposes of time. But then we end up with a very large number of different versions of the DNA that encodes this protein surrounding the virus, is that protein is the delivery vehicle. That's what we're trying to engineer. So we then package these into very large libraries where every single particle is now composed of a variant protein shell that might be the solution to the problem you're trying to solve. And it contains inside of it the DNA that encodes that protein shell. So the particles are effectively barcoded with their own DNA. So let's say you're trying to solve that problem of doing an injection into the bloodstream and having a virus that localizes to the liver so you can treat a heart disease. And maybe this particular variant is the perfect one. You still need to pull the needle out of the haystack. So we then perform the second step of Darwinian evolution, which is selection, where we do an injection, for example, into a non-human primate of the entire library in a single syringe, isolate heart tissue, use molecular biology techniques to pull out the sequences that made it to the heart. We can repackage those into virus, re-administer it, and go through iterative screens so that we end up finding the needle in the haystack. And then we have the variant that excels at being able to carry out the task you're asking of it, which is to carry a DNA medicine to the heart. So I'd like to talk about some of our historical efforts in the field or in this area, and then discuss our efforts to be able to get it into humans. And then talk about improving other facets. I probably won't get to this due to timing, but talk about efforts to be able to improve other challenges within the AAV delivery field. Okay, so first of all, I want to talk about retina and brain. So this is your retina. And if you take a look at what retinal tissue looks like, you have on the back of the eye painted a number of layers of neurons. There's liquid humor or gel inside this, inside the sphere of the eye. And then all the way at the back of the eye are the photoreceptor neurons. These are the cells that sense photons. Unfortunately, mutations in over 200 genes can kill off these cells, resulting in blindness within patients. Think about retinitis pigmentosa, for example. Or more complex diseases like wet or dry age-related macular degeneration can also kill off these neurons and rob people of their vision. So it'd be terrific to be able to get a gene medicine all the way in the back of the retina to this photoreceptor layer. The best way, the best ideal route of administration to deliver DNA would be through what's called an intravitreal delivery into this liquid or this gel fluid on the inside of the eye. And it turns out that this is actually done millions of times a year in the United States. Patients who have macular degeneration have ultra high levels of this protein called VEGF that stimulate the growth of blood vessels into the eye, and that aberrant growth of blood vessels is what causes blindness in wet AMD. So clinical ophthalmologists will fill up a syringe with this protein called Eylea, and Eylea will bind to and neutralize this VEGF, this drug target. And again, this is very, very safe, done millions of times a year in the US. It's about a 4-second injection. Clinical ophthalmologists will do about 50 of these in the morning. So it's well tolerated. Unfortunately, these patients have to get 10 injections a year because the protein has a turnover, but at least the route of administration is quite safe. Unfortunately, if you take a natural AAV and put it into the vitreous, it encounters 200 to 300 microns worth of packed bed and is unable to transport through this dense tissue barrier and reach the photoreceptor neurons. So as a result, in that FDA-approved product where I showed you the movie, investigators had to invent a new surgical route, a so-called subretinal surgery, where they take the needle, and this is really cringeworthy, poke it all the way through the retina and squeeze the fluid containing the virus between the photoreceptors and the underlying cell layer known as the retinal pigment epithelium. So this is by definition a retinal detachment, and the detachment takes a few months to recover from. Sometimes it does damage permanently that compromises vision within patients. That's not a problem with the virus, it's a problem with the surgical procedure. And in addition, this is image, what's called a fundus image. This is what your ophthalmologist sees when they're looking through that lens at the back of your eye, so that you can see vasculature here. And unfortunately, if you puncture through the retina and squeeze liquid, you can't really control how far laterally it goes away from the injection site. And typically only around 5% or less of the surface area of the retina receives the virus. So in that FDA-approved product, it was good enough for them, but they're sacrificing 95% of the retina to the disease. So we wanted to ask if we could do better. Specifically, can we take this library, administer it into the vitreous of the eye, wait a couple of weeks for the virus to penetrate all the way to the photoreceptor layer, isolate the photoreceptors, amplify the virus that made it there, re-administer it, and go through this iterative screen so we can pull the needle out that is really good at being able to make the photoreceptor neurons from an intravitreal administration. So we initially did that in mouse, and I'll talk to you a little bit later about our efforts to translate it towards clinical development. But this is initial kind of a proof of concept within mouse. So we did exactly that experiment, took the entire library, put it into the vitreous, waited for it to transport, or a fraction of the viruses to transport to the photoreceptors, pulled them out. In this case, we're using photoreceptors that glowed in the dark green due to the strain of mouse that we used. So we could use a fluorescence-based cell separation technique to pull out the photoreceptors, amplified the virus that made it there, re-administered it, went around 6 times. And at the end of that, our large library converged down to just a small number, as you can see right here, 4 variants, with one dominant variant that made up the majority of the pool. So we took a look at this variant, and you can see here it contains diversity, shown here in green and also in red, which you can barely see, changes in amino acid composition on the surface of the virus in a position that could change the way that that virus interacts with cells and with tissue. So we then tested its ability to mediate delivery. This is the parent virus called AAV2. This is the variant that's being used in the FDA-approved gene therapy. And if you administer it into the vitreous of the mouse, you can see why they had to do that subretinal surgery. The virus barely penetrates. You end up with a few neurons here and there. So when administered at the same dose, the variant that we created, which we call 7M8, is able to hit every single cell type across the entire retina. And furthermore, we're not compromised or limited to just 5% of the surface area of the retina. We're hitting, the entire retinal surface area, all around, all the way out to the lens or to the margins of the eye. So we then began to utilize this to treat animal models of human disease. This is a monogenic disorder where you can pin the blame for the disease on mutations within a single gene. It's called retinoschisis. It results in loss of this 200 amino acid protein that the protein plays ordinarily a role in causing cell adhesion inside the retina. So in the absence of this protein, the retina falls apart and forms these cavities or schises, hence retinoschisin. And as you can see in this cross-sectional image of an 11-year-old boy who's going blind. So this protein is ordinarily secreted from photoreceptors. So you need to get the DNA medicine there to be able to treat the condition. So this is the mouse model. This is the ERG, electroretinograms. You flash light at the animal, you measure current through the eye. That tells you something about visual function. And typically when you flash, you end up with this so-called A wave, which is due to photoreceptors. And then these are due to the other neurons. And this is a typical wild type ERG response. In a retinoschisis animal, where you intentionally mutate the retinoschisin gene, it goes flatline, which is indicative of the fact the animal is going blind. When we load the correct DNA into our engineered virus and administer it to that mouse, you can see shown here in the solid black line, there's recovery almost all the way back to normal within these animals. And importantly, the extent of that recovery persists throughout the entire life of the animal. And that contrasts with a number of negative controls, including our engineered virus carrying an inert gene, several other viruses engineered by others in the field carrying the therapeutic DNA, which all statistically overlap with animals that didn't receive an injection at all. Okay, so that shows that we can evolve for better delivery, better penetration through tissue within the retina. So what about other tissues? Is this a broad enough platform so that we can utilize it to be able to do therapy in multiple cell targets and multiple tissues? So we moved on to the brain and spinal cord, and the problem is that these are really big tissues. The eye is tiny by comparison. Try to get your bias to distribute through 10 inches of brain. It's really tough to do so. And investigators ran a clinical trial for a pretty tragic disease called Sanfilippo B, which results in the loss of kids. They die before the age of 10, and it's due primarily to loss of expression or loss of a functional copy of a gene encoding an enzyme within the patient's brains, primarily. And so they essentially treated the brain like a pincushion, drilled 8 holes, did 16 injections, space-filled the virus as much as they possibly could, restored less than 10% of natural levels of the enzyme, and didn't modify disease progression, unfortunately. So we asked the question, are there other ways to get transport within this large tissue? And we noticed a number of years ago an interesting property, which is that neurons have a cell body, and then they have a process, a neurite, and then they have terminal synapse at the very end. And if you deposit the virus right near the terminus of this axon, some of it will actually get internalized by the cell, transported back to the cell body, and end up in the nucleus. So you could infect a cell from a distance. So that could be a really useful property. For example, you could use it to sneak virus past the blood-brain barrier. Do injection into muscle. Our spinal cord neurons project to the muscle, our motor neurons, and so they could take the virus back to the cell, to the body of the cell. And a colleague of mine ended up using this to express therapeutic levels of a protein in an animal model of amyotrophic lateral sclerosis, Lou Gehrig's disease. A company tried to translate that to the clinic in San Diego, and they did the math, realized that this process of transport, retrograde transport, was so inefficient that they couldn't possibly scale it up to humans. It became prohibitive. So it's not surprising. These viruses are respiratory viruses. You show it a neuron, it says, what is this? Evolution did not prepare me to infect these cells. So we're lucky that it does it all, and certainly not surprised that it does so inefficiently. So we decided to evolve the virus for this process of retrograde transport. And the way it worked is that this is a mouse brain. Again, this is done as a mouse proof of concept. This is the cortex of the brain. Lots of these neurons send their projections to a single little spot. So if we wanted to infect the entire cortex, we'd have to treat it like a pincushion and do lots of injections. But with a strategically placed injection right here in the pons, the virus can get retrogradely transported back to large volumes of tissue and solve our transport problem for us. So we took the entire library, injected it into the pons, isolated cortical tissue within the mouse, amplified the virus that made it there, reapplied it, etc. And after 5 rounds of selection, we got another variant. And upon injection into the pons of a DNA that encodes a fluorescent protein again, we found that we were lighting up the entire cortex with a single injection. And so we quantified the efficiency of this transport. And shown here is the parental virus, AAV2, and you can't really see it, but there are a couple of green cells there. This is another virus that was engineered within the field. And this is our variant, which shows nice spray painted smattering of very large levels of neurons that are being transduced with this virus. And if we quantify the efficiency, it's actually 50 to 100-fold higher with our retrogradely transporting virus compared to natural versions of AAV. So we could then do that experiment where by strategically placing the virus in just a couple of spots within the brain, we did injections into 3 regions. My students went a little bit crazy with fluorescent proteins, used a red, a blue, and a green fluorescent protein, and showed that with just 3 injections, you could get spread throughout the majority of the brain. So you're using now a different mechanism for transport, this active transport as opposed to passive transport from the retina, where you're getting spread throughout large regions of tissue. Okay, I think I will skip this for the purposes of time. So we had been doing this initially within cell culture and then within mouse models as a proof of concept, and became very interested in doing it for real, getting it into the clinic. And so to be able to do this, about 9 years ago, I co-founded a company. Since companies are the entities that run clinical trials that get drugs approved by the FDA. And this company is called 4D Molecular Therapeutics. We went public a couple of years ago for folks who are interested in that kind of thing. But more importantly for us, that meant that we could capitalize the company to the stage where we could really get into the clinic. And so we have currently 3 different therapeutic areas. I'll show you the retina and the heart. And in addition to the lung, we developed highly evolved variants in non-human primate with the idea that Francis Arnold says evolution teaches you get what you select for. We wanted to select for something that could go into a human primate, so we did all the engineering in non-human primate. We now have 5 ongoing human clinical trials. In general, the technology we've created involving engineered viruses being used in human clinical trials. But I'll tell you about a couple of these, and 3 so far have reported data. In general, of the 8, 5 have reported data to date, and all 5 have been positive. Okay, so we redid the evolution in non-human primate now instead of mouse. We got a different variant, which justified the fact that we should be going into primate. And let me show you its delivery properties. So far, a couple of negative controls, that if you simply inject the virus into the fluid of the eye, the vitreous, it doesn't really penetrate very far in the retina. This is now non-human primate tissue, cynomolgus macaque. And is there a question? Okay, no worries. So you typically only get a little bit of delivery to a small patch of tissue where the barriers kind of thin out a little bit. But this is not good enough for therapy. If you do the subretinal surgery, you get random flow or random directional flow of the virus. You get this weird shaped body of cells that are expressing. But if we use our engineered virus and deliver it into the vitreous of the eye, we get very high levels of expression that turn on pretty soon. And this is an animal that 6 months after administration has glow in the dark, really bright levels of this marker protein. And we sectioned literally through thousands of these sections. And sorry that the color isn't showing up that well. But we find out that regardless of whether we're looking at a bright white section or a lighter gray section, this is simply auto adjustment in the camera. We end up with GFP positive cells or cells expressing the marker protein throughout the entire retina.
Delivering here is only to the vitreous, or are you also doing it to the retina?
No, it's only into the vitreous fluid. So very, very simple administration. Clinical ophthalmologists make money, lots of money doing these injections, can do 50 in a morning. Okay, so we wanted to see whether or not we could get efficacy in a primate prior to heading into human beings. And so let's go back to wet age-related macular degeneration. This is a situation where patients have really high levels of this protein VEGF, which causes ingrowth of blood vessels into the retina and kills off photoreceptor neurons as a result. So I mentioned to you that clinical ophthalmologists administer this protein called aflibercept a few slides ago. And unfortunately, because the protein only lasts a short period of time, patients have to go back into the clinic and get 10 injections a year. And while a single injection is not too onerous, try getting 10 injections in your eye every year for the rest of your life. That's not something patients want to do, patient compliance drops, patients go blind. So we ended up taking our virus and loading into it, not the protein of aflibercept, but the DNA that encodes the protein of aflibercept. So we kind of vectorized this therapeutic. And we also had a second payload that would knock out another gene that's associated with this blood vessel growth. So we then took a non-human primate, and there's a model called that causes vascularization. You get a little bit of a laser burn, and it causes blood vessels to grow within the eye. And so you can administer a therapy and see if it blocks the growth of these blood vessels. And we did that for 6 months in life. So first off, at a very low dose of the virus, we can get detectable levels of this aflibercept within the eye. In addition, we quantified the incidence of what are called grade 4 lesions, serious growth of blood vessels after the use of this laser. And in a vehicle control, we end up with a large number of these lesions. In any of the doses we tested, 0 lesions. So the therapeutic was able to completely block the blood vessel growth within the eye. And in addition, it resulted in a decrease in the size of the blood vessels or the lesion that was introduced. So based upon the strength of the non-human primate data, we began to put together IND packets, investigational new drug applications, to ask the FDA permission to go into phase 1 human clinical trials. And we've done so in the retina so far for 3 diseases. And I'll describe one of them to you. So this is retinitis pigmentosa. This is a blinding disease that robs young boys of their vision. It's called X-linked retinitis pigmentosa. And what happens in RP is that initially you start out with a full field of vision, and the peripheral vision begins to die off. And then the patients, unfortunately, are looking through the world, at the world through a straw. And then finally, the last bit of vision blinks off, and they're completely blind. So that can be reflected, or you can see that actually here in the area of the retina, which starts out relatively large. In this form of RP, by age of 20, patients are essentially blind. They've lost most of the surface area of the retina. So in our clinical trial, we used 2 endpoints, 2 clinical measurements. The first of these was exactly this measurement. You can do an imaging technique to be able to measure retinal thickness or retinal area and watch it decrease over time, unfortunately, in patients who are untreated. And this is a functional, think ERG, kind of functional test of whether or not somebody can see light. So unfortunately, in a phase 1 trial, the FDA typically only allows you to treat patients who are late stage, so they have nothing to lose. So we're treating patients that are out here, but we're moving into a phase 3 clinical trial that will now enable us to treat patients who still have retina to protect. But in 6 patients that have been treated so far, 2 of them had measurable retina left. And in both of these patients, we saw a slowing down in the rate of loss of the retina and an improvement in visual function and light sensitivity. So based on that, we're currently moving to a phase 3 clinical trial. And in all, we have 3 products into the clinic now in the retina. So there's X-linked retinitis pigmentosa. This is a disease called choroideremia, which is similar. It robs young men of their vision, typically by the age of 30. We've seen improvements in the function of these retinas and vision. And in addition, wet AMD, where we treated patients for the first time a bit earlier this year, and we'll be reporting out data probably within a couple of months by the end of the year. We also engineered viruses. This one has the profile, if you inject it into the bloodstream, it goes to the heart. So we're using it to treat a condition called Fabry disease, which I'll show you in a second. And then in addition, in this case, we took virus, aerosolized it, had people inhale it, or animal subjects, and then isolated lung tissue and amplified virus that made it there. So this is cells that carry DNA into the lung. And so we actually initiated a clinical trial several months ago for cystic fibrosis, but we're far away from data in that case. The other trial where I wanted to show you data is Fabry disease. This is a disease, again, caused by loss of function mutations within a particular gene that encodes an enzyme that's important for cardiac function. And these patients typically die as a result of cardiac disease. So we took the DNA encoding the normal copy of the gene, put it into the virus, administered it intravenously into patients, and then took a look for an increase in the level of expression or production of that enzyme, which is otherwise lost within those regions. So this on the y-axis is the levels of the enzyme. Shown here in blue is the normal range of enzymes within the human population. And at the low dose of this virus, we accidentally overshot. So we're producing more therapeutic than what the patients would need to eliminate the disease. And it's perfectly safe. There's a recombinant protein therapy where people will get even much higher than this. It's entirely safe. But this is a situation where we're getting delivery to the heart, which the recombinant protein doesn't. And it's one injection potentially for the life of the patient. This patient was a little bit different. The patient, unfortunately, took the recombinant protein, developed antibodies against it that kind of accidentally vaccinated the patient against the protein or the enzyme. So that extremely high levels of antibodies against the therapeutic protein, but we're still able to treat through that and achieve therapeutic levels of the enzyme. And so we're continuing to monitor these patients. Okay, so in the next 5 minutes that remain, I'd like to talk about some other challenges that we're beginning to tackle within the field. If you can't deliver your therapeutic, there is no drug. Stop there. There's no point in continuing. But as we begin to feel comfortable in the way we're chipping away at delivery, other problems start coming to the fore, and in particular, manufacturing, which is, of course, something that's near and dear to chemical engineers, and the field is going to need lots of chemical engineers. So the way that the manufacturing works right now is that, unfortunately, you need to produce this virus in cells that adhere to a surface. So you have these producer cells, you put pieces of the viral DNA inside of it, cell processes these, and spits out viral particles that contain your gene of interest. And right now, there's a major manufacturing bottleneck within the field. If we think optimistically, we can start treating some of these diseases, that there's a bottleneck within the field. So I described to you several monogenic conditions where we're talking about 20,000 patients in the US and Europe. The retina is a really small tissue. That's not going to be that hard. Wet age-related macular degeneration, 1 million patients in the United States. So that's going to get tougher to manufacture. As we head towards other diseases that affect the heart or muscle, we're going to need massive doses of the virus, and the field, quite simply can't produce enough. So, for example, that FDA-approved product, spinal muscular atrophy, there's a certain annual incidence of patients who are born with SMA, and it requires a certain dosage of the virus. So to be able to manufacture enough virus just to treat that incident population, 20,000 square meters. Just to calibrate, a soccer field, a football field is around 5,000 square meters. So we're talking massive surface area. There's a therapy that is under development for Duchenne muscular dystrophy. There are lots of DMD patients. It requires a massive dose of the virus because it's not very efficient at being able to reach muscle. If we can make a targeted vector for muscle, we can alleviate the manufacturing burden a bit. But right now, we're talking 7.5 million square meters of surface area to produce enough virus to treat the current population. So this rose up to the level that your hometown newspaper, the New York Times, back in 2017, published an article talking about the bottleneck in manufacturing in the gene therapy field. Okay, so since I'm from Berkeley, I'm contractually obligated to use CRISPR. So I'm going to talk to you about some work in which we did genome-wide screens with CRISPR to be able to identify changes to producer cells that result in better manufacturing of the virus. Okay, so CRISPR, as you likely know, it's a protein. You give it a little bit of an RNA guide, where that RNA is, let's say, a Watson-Crick base pair match for a specific target within the genome. The CRISPR will use that guide, or the Cas9 will use that guide and go looking for the Watson-Crick base pair match, and then Cas9 will drag with it whatever cargo you might want to carry for the ride. So, for example, the typical way in which it's used is, Cas9 is a nuclease. If you target it to a gene in the genome, it'll cut that gene, the cell will make some mistakes in repairing that lesion, and it'll knock out a gene. So using this, you can knock out in principle any gene in the genome. The other way in which you can use it is to create a Cas9 that doesn't have the ability to cut DNA, but where it carries with it proteins that turn on a gene. And again, you can design a library of guide RNAs that will carry this to every single gene in the genome. So you have the ability to either knock out every gene in the genome or turn on every gene in the genome. And the guides that are used to direct where these systems should go, we took these large libraries, and in particular, this one has 70,000 guides in this library, so 3 guides for every gene in the genome. And this one has around 23,000 or so. The knockout library has 58,000. So we took these guides and stuck them into the viral DNA. And the idea is that we want to find genes where if you turn them on really strong or shut them down completely, resulted in better manufacturing or better production of the AAV. Okay, so we packaged this. So now we have a library of particles. Every single one is carrying a different guide RNA. We can then take these, infect our producer cells, and wait 48 hours for them to either turn on a gene or shut down a gene. And then we introduce the other little pieces of DNA that the virus needs to package. And we now, these viruses, with the help of these constructs, replicate. So we've now produced, gone through one round of selection. So if this guide RNA, let's say the one shown here in red, in this red virus, resulted in an increased production or expression or turned on a gene in a way that modified the cell such that it could better produce AAV, then there are going to be more copies of this red virus that are produced from those cells, right? It'll generate more. So you can take these, reinfect the cell, go through another round, and through successive rounds of selection, you end up increasing in prevalence the frequency of the guide RNAs that are beneficial for viral production. So it's another selection system, another Darwinian selection. So we could then sequence those guide RNAs, and sequencing them will tell you what gene they're directed against, again, because of the Watson-Crick base pairing. And we could sequence them before versus after the selection and look for an increase in frequency. So you can see this particular variant increased in frequency almost 3,000-fold as a result of doing these selections. So this, probably really encodes a guide RNA that really tells the cell to crank out the virus. And so we identified the target genes and then asked the question, are we increasing viral production from these cells? So when we introduce these guides singly, we find around a 2- to 3-fold increase in viral production. But we then got the idea of, doing it combinatorially. And then we're getting a 5- to 10-fold increase in viral production. So in other words, we could potentially go from 7.5 million square meters down to 750,000, which is still a daunting problem. But I think a 10-fold increase in production is something that a chemical engineer might be enthused about. So I'm going to skip ahead a little bit and skip past the third story and jump right to the conclusions for the sake of time. And say that genetic diversifications and library screens and selection are a very, very powerful approach to find solutions to complicated biological problems. And even though I didn't describe it, we've actually, in each one of those situations, taken the solution to the problem, reverse engineered it, so we can understand something more about mechanisms, something more basic about why that virus delivers better or why that cell produces more vector. So we've used this approach to reprogram the soccer balls to get variants of the virus that have enhanced efficiency, targeted delivery, and in work I didn't talk about, new versions of the virus that have the capacity to evade the antibodies that most human beings have against the natural versions of the virus. We're happy that these are advancing or have advanced into the clinic, and we're now tackling next generation problems now that we feel good about what we're doing with delivery, and have used this to be able to chip away now at the manufacturing problem and result in improvements in process that we think are going to be meaningful as we go to the clinic further. Okay, so I'd like to acknowledge the people who actually did the work. The work in the mouse retina was the product of Leah Byrne and Denise Kara, who are both PIs of their own labs now, in collaboration with John Flannery at UC Berkeley. The work on that retrogradely transporting virus within neurons was the product of Tom Guy and Bumyol Wong. We did this in collaboration with Genelia Farm, Alla Karpova. The work on generating variants in vivo and taking them into the clinic, I'd like to acknowledge our company team. So David Kern, my co-founder, Melissa Cotterman, who's since left, but she was SVP, and especially Peter Francis, who's both CSO as well as CMO of the company. And finally, the work on improved production of the viruses was the product of Chris Barnes as well as Huncho Lee. So thank you so much for your time and attention, and I'd be happy to address any questions.
Hiring adventure. Would like to have the first question? Yeah.
So thanks for that. So looking at the viruses that were eventually selected, like the needle in the haystack, can you say anything about what makes them more like efficient, and is it specifically for each body region, like tissue, or is it specific to a disease or a function? What is the?
Yeah, it's great. It's a great question. We always want to reverse engineer and understand mechanism. Sometimes, we don't. Our first priority is get it into patients, and then the second, I'm really interested in understanding more, about how this biology works. So the one that we know the most about is the retina one, and there were two changes. If you remember that cartoon, there was one in red and one in green. So these viruses typically latch onto cells by binding initially to a sugar receptor on the cell. And then they bind to a protein receptor and they get internalized and trafficked through the cell. So this particular one binds to a sugar that's present not only on the cell surface, but it's also present a lot within just the matrix of tissue. And this is a 20-sided dice or a 20-sided soccer ball. It has 20 binding sites for that sugar. So what happens is it has greater than picomolar affinity, sticks like glue to the sugar in the very first layer of tissue, and doesn't have any ability to actually diffuse through the rest of the tissue because it's stuck. So it's, what chemical engineers would call an absorption, diffusion, or convection problem. So it spends too much time absorbed and not enough transport. So one of the mutations actually decreased the affinity for the sugar, so you might imagine you can go through a Brownian process where you bind, unbind, take a Brownian step this way, rebind, unbind, take another Brownian step. And so it has the capacity to penetrate through the tissue because it spends more of its time unbound. And then the second change enabled the virus to infect cells to bind to a different receptor on the cell surface to gain entry into the cells. We haven't identified what that receptor is though, but it's new information. It changed the function of the virus.
So it is tissue specific.
Yes, those three variants that I showed you for retina, heart, and lung, completely different sequences.
And what was the initial size of the library, like the full?
It's over 1 billion synthetic versions of the virus now. Thanks. Yes.
Hi, I have a question about, you mentioned in one of your slides a viral vector that had 2 genes that it was delivering, and I've just never heard of that before. Is there any kind of transport or efficacy limitation caused by putting too much DNA into the viral capsid, and how does that been engineered?
Yeah, you run into I mentioned in the intro slide that this is the smallest or among the smallest of viruses. It only has about 5 kilobases worth of real estate to fit DNA inside of. So the average size of a protein encoded in the human genome is only around 60,000 molecular weight. So you can actually fit 2 of those into these viruses. However, that size distribution of proteins has a long tail, and above some threshold, quite simply can't fit it into the virus. So we've been addressing that problem in work I didn't touch upon that much, but maybe I'll show you a little bit. So the DNA has a couple of elements in it. One is the coding sequence, and the other one is kind of a controller sequence that determines how much messenger RNA is synthesized. It's called a promoter. And that promoter typically takes up some real estate. It takes up 600 to 1,000, maybe sometimes 1,500. So it's stealing space away from the coding sequence. So in this case, we were doing evolution through libraries of these promoter elements to find really strong, tiny ones that are so we created one that is this is the promoter we took into the clinic. So it's 1,600 bases, so really long. It steals a lot of real estate. If you wanted to fit a large protein, you couldn't do that along with this one. So these engineered promoters are now about 1/5 of the size of this large natural one, but are just as strong in producing messenger RNA. So that's one way in which you're trying to solve the size limitation issue.
Are those smaller promoters that you found, are they performed by directed evolution?
Yeah. Yeah, we create large libraries of candidate promoters and do selections for the ones that are the strongest. Oh,
based on the size of this library, this makes me think that you could use some sort of neural net or artificial intelligence modeling program to kind of predict the best viral candidates for the future diffusion, the better candidates to promote this transport. Have you tried modeling any?
You just described the paper we submitted 5 days ago. Yeah, yeah, So sequencing now, next generation sequencing where you can sequence, like a dozen human genomes in a week on a single instrument. What it gets you is tons and tons of short little reads, so 300 nucleotides, but you get like 8 billion of them. And so in some situations, the diversity in our library can fit, we're making a targeted change, a random change to a specific region of the virus, and that sequence can fit inside of 300 nucleotides. So we then take that library, let's say we're doing the heart problem, where we inject it into the bloodstream, we're pulling out the heart, and we can sequence what was in the syringe, and then we can sequence what made it to the heart. And that gives you, for 100 million variants, or 50 or 100 million variants, it gives you the relative efficiency of each one, because if it's more efficient, it should show up more times in the heart, right? Higher number of copies made it to the target site. So we use that to train neural networks so that we can make them develop predictions about variants with improved properties. And that was really useful because this protein is 735 amino acids. 20 amino acids raised to the 735th power is greater than 10 to the 900th. So we're, most of those are not going to fold into a soccer ball, right? But we are very sparsely searching space. But with a neural net, with a machine learning algorithm, you can start making predictions about the properties of variants that weren't even in your experiment. And so that's a direction we're very much going.
Maybe one more question. Okay, great. So with this, we'll thank Dave again.